Hello friends, warm welcome to all to the channel World of Zoology. In our today's lecture, we'll start with the next point, that is application of biotechnology. And in that, we'll consider first application, that is DNA fingerprinting, a technique in brief and its application in forensic science for investigation of the crime. In the past, criminal investigations and trials heavily relied upon forced confessions and witness testimony. But solving the criminal cases in foolproof manner has become possible due to the forensic science and its progressive development in various techniques for crime investigation. One such technique is DNA fingerprinting or DNA profiling, with the help of which forensic scientists and legal experts can solve crimes, defend legal suits, etc. It is an investigation procedure that basically distinguishes individuals with the help of their DNA analysis. This genetic analysis is based upon identifying tiny segments of the hereditary material from a biological sample left at the scene of the crime that can be analyzed and compared with the DNA of likely suspects. In addition, DNA fingerprinting can be used to determine whether individuals have been wrongly convicted of a crime. In other instances, DNA analysis help to determine paternity and identify victims of disasters or accidents, like establishing the identity of burned or unidentifiable dead bodies. In 2005, the Criminal Procedure Code 1973 was amended to allow the medical practitioners, after authorization from a police officer who is not below the rank of sub-inspector, to examine a person arrested on the charge of committing an offense and with reasonable grounds that such an examination of that individual will bring to light evidence regarding the offense. Section 53, as amended, includes the examination of the blood stains, semen, swabs in case of the sexual offenses, sputum, sweat, hair samples, and fingernail clippings by the use of the modern and scientific techniques, including DNA profiling and such other tests which the registered medical practitioner think necessary in the particular case. Technique of DNA fingerprinting will consider next. Sir Alec John Jeffrey, a British genetist, was pioneer in developing the technique of the DNA fingerprinting in 1984. This method has also been patented and is practiced in Europe and America. It is based on the use of mini satellite DNA. These are short sequences of DNA that differ from person to person, though almost 99% of bases are same in the DNA of all human beings. That means the genetic material of any two person, it match near about 99%, and only the 1% unmatched can show the individual difference. Out of 1 in 1000 base pairs of the total human DNA is a site of variation of such mini satellite DNA sequence that is about 10 to 15 base pair long. These short stretches of sequences are also repeated several times along the entire DNA and hence are referred to as variable number of tandem repeats, VNTRs. variable number of tandem repeats, that is VNTRs, means the short stretches of sequences of the DNA and which repeats several times along the entire length of the DNA. These hypervariable sequences or segments of the DNA are first isolated and identified by White in 1918 at the University of Utah. Since the length of VNTR differs from person to person, similar to the fact that the fingerprints of every individual are different from the another person, it becomes the basis for establishing correct identity of that person depending upon length of VNTR. On the basis of number of repeats present in VNTRs, the length of segment is measured. In different persons, this length differs and therefore VNTR is the key of the DNA fingerprinting. Because of this extensive variability in the length of the sequence of the mini-satellite DNA, the chance of finding two individuals in the population with the same DNA fingerprint is about 1 in 10 raised to 5 to 10 raised to 6. Sir Alex Jeffrey developed the method for measuring the length of VNTRs, which is also known as a restriction fragment length polymorphism, and in short RFLP, that is restriction fragment length polymorphism, RFLP. In every individual, one set of mini-satellite DNA sequence is inherited from each parent in human mini-satellite DNA sequences. Therefore, an individual's DNA binding pattern based on the mini-satellite DNA sequence is almost as unique as his or her fingerprints. Hence, the method has been referred to as DNA fingerprinting. Generally, there are two strategies for determining DNA relationship among humans. One approach that depends upon the DNA hybridization to under undegraded mini-satellite DNA. Alternatively, with minute samples with degraded DNA, a set of polymerase chain reaction, that is PCR primers, can be used to determine whether two samples have the same amplified DNA band. How the method works, that is RFLP method. For mini-satellite DNA typing, the sample DNA is digested with a restriction enzyme and the fragments are separated on agarose gel and transferred by blotting onto the nylon membrane by southern blotting method. The membrane is hybridized sequentially with 4 to 5 labeled probes of known sequence. Probe means labeled sequence of the DNA. And each probe recognizes a distinct DNA sequence. After each hybridization reaction, the bands where the probe has bound to the digested DNA sample are visualized by autoradiography, and the banding pattern for each sample is noted. Before next probe is used, the first probe is completely removed from the membrane. Since each hybridization and autoradiography step can take up 10 to 14 days, the entire process may take many weeks and several months. The commonly used set of probes of human mini-satellite DNAs have been standardized for this technique. In India, facilities of DNA fingerprinting are available at the Center for DNA Fingerprinting and Diagnostics, Hyderabad. At this center, the Jeffries technique is not followed. Dr. Lalji Singh has developed an indigenous technique where BKM DNA probe is used for hybridization of DNA sequences. While he was working on sex determination in snakes in his Ph.D. program in BHU Varanasi, he found a contrast result. Unlike human and other organisms, the female snake contains XY, whereas the male has YY sex chromosomes. A segment of DNA was isolated from sex determining Y chromosome of the female banded crate. This unique segment is similar to sex determining chromosome in humans and is named as banded crate minor satellite, that is BKM. This segment is used as the probe and is called BKM DNA probe. Based on the quantitative differences in the copy number of BKM related sequences in the given sample of DNA, the exact profiling can thus be done for further investigations. Application of DNA fingerprinting technology, that is the next point of our lecture. It is very sensitive technique, being able to generate sufficient data from only tiny amount of even partially degraded biological material. It has far greater power of discrimination between individuals, sometimes up to 1 million fold higher than conventional techniques. This technique has application in areas like forensic science for crime investigation, medical diagnosis, and parentage testing, setting up of genetic data bank for application in the field of agriculture and animal husbandry. It is of great significance in the study of evolutionary relationship of the animals. This is presently being used for wildlife preservation and better management of endangered species. One of the application of DNA fingerprinting in crime investigation is infamous late Rajiv Gandhi assassination case of 1992. Establishing the identity of Danu, the suicide bomber who killed him, was done by using DNA probe. Equally crucial was comparing the DNA of the mastermind behind this assassination case, uh, Shivarasan's DNA, with that of his parents who were flown in India from Sri Lanka. This is regarding DNA fingerprinting, that is the first application of the biotechnology. Thanks for watching this video. Do like if you like the lecture. Share this video for the benefit of the maximum students and subscribe the channel World of Zoology for the notification of my next video. Thank you once again.